Okay, so for those of you who were in the lecture, you realize that I forgot to record it. For those of you who are now just listening, I'm going to go through and redo what I did for your classmates because I forgot to record the actual synchronous lecture today. So let's go ahead and just walk through the questions for today. Remember, learning outcomes for chapter 26 are here. A lot of chapter 26 you guys have seen being played out in real life right now with all of the COVID-19 stuff. What that means is that some of these terms actually might be more familiar to you than they may have been beforehand. A lot of epidemiology is understanding the terminology, but then it's also doing the calculation. So we'll practice some of those today. Okay, so let's take a look at this question here. Okay, it says last year the incidence rate of disease A was 30 per 100,000 for month 1, 40 per 100,000 for month 2, and 35 per 100,000 for month 3. Keep in mind, incidence rate means we're talking about a proportion of a population. And we care about rates in epidemiology because when you look at the percentage of a population, it puts it into perspective as to how much of that population is being affected. And often we will express those as 30 per 100,000 as opposed to, say, 0.5%, because people don't really picture what 0.5% is, but if you tell them that 30 out of 100,000 people, they can picture that a little bit better. And then we've got more terminology here. Incidence rate is the number of new cases. So when we say that this is 30 per 100,000 for month 1, it means there were 30 new cases of disease A during that month. Typically, the incidence rate of this disease ranges from 10 to 15 per 100,000. Then we want to know what this tells us about the disease. So A says there's an epidemic of disease A because the total number of cases were higher. B says disease A was endemic because the number of new cases each month exceeded what was expected. C says it was an epidemic because the number of new cases exceeded what was expected. And D says disease A was endemic because about the same number of people would have been died. So again, more terminology, the difference between epidemic and endemic. Endemic are the baseline levels. What do you normally expect of a disease? And in this question, that's the 10 to 15 per 100,000 because it tells us typically the incidence rate of this. That would be the endemic expected level. 30 to 40 is 3 to 4 times higher than that, which tells us we're having an epidemic, a higher than expected number of the cases. And we already said incidence rate were new cases. Okay, so we're going to want to look at this, and this is going to be C. This was an epidemic of disease because the number of new cases each month exceeded what was expected. And so that's making sure you understand the terminology, but not just that you can define things, but you can use that terminology. Okay, this didn't just say define an epidemic. It's being able to understand that this is a higher number of cases, so it is an epidemic. Okay, so we mentioned incidence rate in the last one. Okay, let's actually look at how you calculate those rates. Okay, so we're working as an infection control nurse in a hospital. There are 300 patients in the unit. This month there were 5 new cases of MRSA, which brought the total number of new cases on your unit to 15. Okay. And it wants to know what's the prevalence of MRSA expressed per 100 patients. So now we're going to look at how to calculate rates, but we still need to know the terminology. Prevalence is talking about the total number of cases. Okay, so the difference between incidence and prevalence is that incidence is the number of new cases. It's really going to be telling you how a disease is spreading through a population. That's why we keep looking at the new cases of COVID-19 every day. We want to see how many new cases are happening, what's the incidence rate each day, because that tells us how fast it's spreading in the population. Versus prevalence, which is telling you how much of your population is expected. So what's the burden on your population that's actually infected right now? So the prevalence rate is going to equal the total cases divided by the population at risk. Okay. And keep in mind that you calculate these rates the same way. So if this had asked for incidence rate, it'd be the same type of math, only instead of prevalence as a numerator, we would have had new cases, we would have had incidence as the numerator, and it still would have been divided by the population at risk. Okay. So in this case, our total was 15, and that should say unit. So 15 cases, and our population at risk are the patients on our unit. Okay, we're not asking about what this is in the community or the state, and so our population at risk are those 300 people that are currently in the unit itself. We do that math, we're going to get 0.05 as an answer, where that is the same as 5%. And so our prevalence rate is 5%. But again, people can't really picture that, so instead we're going to express it per 100 patients. So we would take our prevalence rate, multiply it by 100, and that's going to give us 5 per 100 patients. Okay. So there's 5 cases, okay, 5 people who have this disease, who have MRSA, for every 100 patients on your unit right now. You would do the same exact math for incidence, only incidence would be new cases, so it would have been 5 divided by 300. That would have given us a number, and again, we would have multiplied it by the per number of patients we wanted to express our value for. Another key difference for rates is understanding the difference between mortality rate and fatality rate. And actually, I updated this on the last one. Let me update it on this one. So if we do this for real time, the number of cases right now is 17,906. And as of this morning, we've had 158 deaths. So let's use the real numbers because this is looking at COVID-19. So right now, we've had a total of 17,906 cases of COVID-19 and 158 deaths since the start of the pandemic. Again, here's another term you should now recognize. Hey, when an epidemic crosses borders of countries and goes to other continents, we then consider it a pandemic because it has gone worldwide. There's currently almost 3 million people living in the state, and we want to know what's the mortality and the fatality rates. So there is a difference between those. They're both dealing with the number of deaths, but mortality is looking at your number of deaths compared to your entire population at risk, versus fatality is the number of deaths from people who have gotten sick. Okay. Mortality is telling you, if you live in this state, what's the chance of you dying from COVID-19? Fatality is saying, if you get COVID-19, what is the chance that you die from it? So if we look at the difference between those, they both have 158 deaths, but the population at risk for mortality is the population of Utah, and the population at risk for fatality is the number of people who actually have been sick. So if we do that math here, we're going to get a pretty small number. But then remember, we're going to express per 100,000 people. So we multiply by 100,000. Okay, that's going to give us 5.7 per 100,000 people. Okay, so 5.7 deaths. Versus fatality, which is going to give us a bit of a smaller number, times the 100,000 to express it. That's going to give us 882.4 deaths per 1,000 infected. Okay. What that means is living in the state of Utah, there'd be 5.7 deaths from COVID-19 for every 100,000 people in the state. So out of every 100,000 people that live here, 5.7 of them will die from COVID-19 with our current data. Fatality says that if we've gotten 100,000 infected people, we can expect 882 deaths from the disease. Okay, this is if you've been infected. Most of the news is reporting it right. So most of the news, you'll hear them talking about case fatality rates. Okay, that's what these are. So if you see anything that says CFR, that's your case fatality rate. But sometimes you will hear people talk about mortality rate when they actually mean fatality rate. If they're talking about the number of people who have gotten sick and died, they're talking about fatality, not mortality. And unfortunately, people tend to use them interchangeably sometimes. Also as a comparison, so if we change this to the actual percentage, this is 0.88%. Okay, so the fatality rate of COVID in this state is almost 0.9%. Your standard influenza fatality rate is about 0.6 to 0.1%. Okay, so right now, the case fatality rate in Utah is about 8 times higher than that of influenza. Okay, so those are the math and the calculations. Now we're just going to go through some of the terminology and the other concepts in the chapter itself. So this says, what is one reason that epidemiologists use surveillance to do their jobs? Hey, all the numbers we've just been calculating, to figure out incidence or prevalence, you have to get that data from somewhere, and that data comes from surveillance. And so, the lab will report a case to the local public health department. A certain disease has occurred. The public health department will report it to the Utah State Public Health Department. The Utah State Public Health Department will report it to the CDC. That's surveillance. And what does it let epidemiologists do? Okay, so A says it lets them determine the endemic level of disease so they can identify epidemics or outbreaks. B says it lets them examine if a specific public health measure is currently effective. And C says this lets them determine what population, what locations are most affected. Okay, the answer to this one is D. All of those above are reasons for public health surveillance. So we don't know if an epidemic is happening if we don't know the normal baseline levels of disease. And so we always are monitoring to know that endemic level so that we can then identify if something's gone wrong and we're now seeing higher than expected numbers. It also lets us figure out if things that the public health department are trying to do are actually effective. So, for example, with social distancing and asking people to wear masks, you can look at areas that are doing that well and see if they have affected the incidence rates, and areas that are not wearing masks and see if that has changed things. And that's how you can use epidemiology and surveillance to study these public health measures and see if they're functioning. And by the way, we do see less incidents. We do see lower spread if people are social distancing and wearing their masks. We've seen that data. It also lets them determine what population and what locations are most affected. And we've seen that if you look at the state right now, with Salt Lake City still being in orange, while other parts of the state are now yellow and even green. Some of those colors might change with our current numbers, but this idea of determining the locations and the populations that are most affected and letting you target your public health measures to where you most need them, we're seeing in real time right now. So everything we've talked about up to here has been what we call descriptive epidemiology, doing the surveillance, collecting the data, and then just looking at what the data means. So what is the data telling you? Hey, how many new cases are there? How many deaths are there from a disease? What areas are those happening in? That's describing what's currently happening in the population. Analytical epidemiology is then being able to look at those descriptive numbers and try to study them. So to try to actually answer questions, why is this happening more often in one population than another? And what risk factors might someone have? Those are then analytical studies, and there's a couple different types of those which we're going to figure out looking at this problem. So this problem says we're trying to identify risk factors for contracting severe COVID-19. Taken a group of people that have severe COVID-19 and a group without. And now they're looking for factors that might be different between these two groups. And we want to know what type of study is this. So I just told you it's not a descriptive study because we introduced it with this is going to be an analytical study. But what type is it? This is one of those things that confuses students sometimes. So we need to see how we've set this up. So we've taken a group of people with COVID-19. So we have a group of people with the disease, okay, and then a group without it. When you group based on disease status, okay, this is a case control study. You're then going to look at both of these groups and compare what risk factors they might have. Okay, what are their traits? What exposures might they have had? And this is what's currently happening right now. And so we've taken people that have severe disease, and our without disease group has actually been people with mild symptoms. Okay, so your controls don't have to be completely without disease. If we want to know really what's leading to that severe COVID that requires hospitalization, we can take those people with severe COVID that requires hospitalization and compare them to those with mild symptoms to start to see what are the differences between those types of people. what we're learning is that we're finding underlying conditions that really might influence your risk factor of getting severe disease. One of the biggest ones we're finding is diabetes. So diabetes or prediabetes is turning out to be a risk factor for severe COVID. Okay, this is different than a cohort study. Okay, so the answer to this one is C. Okay, a cohort study usually will come after a case control. So now that we are realizing that diabetes might be a risk factor, what a cohort study would do is it groups based on the exposure or the risk factor. So we would take people with diabetes and people without diabetes, and then we'll follow them for a couple months to see who develops disease, okay, who develops COVID-19. And if diabetes is a risk factor, we would expect to see higher numbers of cases here than we would over without the diabetes group. And this is a cohort study because we've grouped based on risk factor and then looked for disease, as opposed to vice versa with case control studies where you group based on disease and then look for risk factors. Okay, and then the last question, wanting to know, okay, up to 70% of newly emerging diseases are zoonotic, okay, because emerging diseases often occur A, when humans go swimming in freshwater lakes, B, when humans come into contact with populations of animals that they typically would not, C, when humans rely too much on antibiotics leading to development of resistance, or D, when diseases with unknown etiology are reported to the public health department. So should remember from the last chapters, so last week, zoonotic diseases are when you have a reservoir of the disease that's an animal. And many of our newly emerging diseases, remember, those are diseases that are brand new to humans, okay, they've never shown up in a human population before, are found from animal reservoirs. And so the answer to this one is B. Okay, most of them are zoonotic because they're really occurring when we're now coming into contact with populations of animals that we typically wouldn't. And this is usually because of human behavior. So this is when we are now expanding into unknown areas. We are cutting down forests and expanding there. We're coming into contact with animals. If we are bringing animals into our locations, like the wet markets in China, we are taking animals from the wild, we're bringing them into contact with new people. Climate change is also doing this because climate change is allowing animals or, say, insect vectors to now live in new areas where they wouldn't have been able to live before. As things warm up, mosquitoes can now live in greater areas. That's bringing new transmission and new diseases into those areas. And so newly emerging diseases, brand new to humans. Okay. D here is important because reporting these unknown etiologies, this is how we identify that there is a new emerging disease. It doesn't answer this question, but this is how we recognize that these newly emerging diseases are happening. If you go in with respiratory symptoms and they test for all the known respiratory pathogens and they can't find one, they're going to report that to the public health department as unknown etiology, because if there's also been 10 other people that have a respiratory disease that they can't find a pathogen to, that's now a group of people with the same unknown etiology that sends up a flag that says there might be something going on here. That's when the researchers and scientists will come in and take samples and start to try to identify, is there a new pathogen we didn't know of in these samples? And you have to have that reporting to be able to identify these newly emerging diseases. When we look at C and it's talking about development of antibiotic resistance, these are what we call re-emerging diseases. So we have newly emerging and re-emerging. The newly emerging are the ones that are brand new. Re-emerging diseases are diseases we know about, but we've had under control. They're diseases we could treat, that we could take care of, but now they're re-emerging. They're coming up in higher numbers in the populations, most often because they're now resistant to the methods we have to treat them. And so this antibiotic resistance is really leading to this development of these re-emerging diseases that are now becoming harder and harder for us to treat. Okay. I mentioned in the lecture when I was giving it live, and this is kind of my baby. So I have a certificate in biodefense and emerging infectious diseases. I teach the emerging infectious diseases class here at UVU. So if this type of stuff interests you, come take that class with me when I teach it. It's a actually really fun class. But I won't go on forever and ever about newly emerging diseases. I will, however, in the live lecture, I did have a question asked about the case study, and it's one that I typically get asked, so I want to answer it. And it was the idea of what type of epidemiological study was being seen in Haiti. Remember, you can have propagating epidemics, which typically will look like this, where you get multiple loops because this is propagating. You're spreading from one person to another to another. So these people are spreading to those people and those people are spreading to those people. Or you could have point source, which goes up and comes down, and all these people were just exposed to one source. We see these with things like foodborne outbreaks where everyone eats the same thing, they all get sick, and then nothing else happens because they didn't spread it to anyone else. The graph in the case study for Haiti looks like this, and it's waterborne. So typically waterborne diseases are most often point source. Everyone who drinks that water gets sick and that's it. But what happened in Haiti is everybody drank from one water source. They got sick, but then some of them contaminated another water source, which then made a new group of people sick, who then contaminated other sources and made another group of people sick. So that is propagating. And in the case study, I say don't pay attention to the heights. Okay, normally disease spreads in the population, the amounts get higher. In this case, they decreased because what happened was public health. Okay, epidemiologists had come in and said, boil all of your water. So even though we had another contaminated source and we spread it, more people were now boiling their water, so less of them got sick. If this had naturally been allowed to happen, we would have seen the standard bigger and bigger as they went, but public health measures worked. Okay, so for propagating, as long as you're seeing groups appear, that is a propagating curve. It is spreading through the population. A point source will always just go up and then come back down. Okay, so hopefully this helps everyone understand epidemiology. Remember, you've got exam 4, and you also have your final reflection as your exam. So a couple big assignments that you've got to do this week. Be sure to reach out if you have questions, and good luck on everything.